Good morning, everyone. Good morning, North America and South America. Good afternoon, Great Britain, Europe, and Africa. And good evening, Asia. My name is Ron Vale, and I'm the Executive Director of Janelia, the research campus of the Howard Hughes Medical Institute. And I would like to welcome you to Life Science Across the Globe, a sister institute seminar series. This series is open to viewers anywhere in the world. And more information on the series is found on this slide, as well as the website lifescienceacrosstheglobe.org. And videos of prior talks can be found on this website as well. Each week we feature two talks. First, a science talk, and second, a talk on science culture. Today, we cross the equator and welcome South America. And I'd like to welcome our third sister institute, the University of Buenos Aires and CONICET in Argentina. And I'd like to welcome Dr. Belén Elgoigian, who was named the 2012 Researcher of the Argentinian Nation. Welcome, Belén.
Good morning, good afternoon, good evening. Welcome to the Life Sciences Across the Globe weekly seminars. My name is Ana Belén Elgoigian. It is my great pleasure to introduce the two speakers for today's seminars. Both speakers belong to the University of Buenos Aires and CONICET from the Argentine Sister Institute of the seminars. They are both among the most outstanding scientists in a region. The first talk will be delivered by Alberto Kornblihtt. Alberto graduated as a biologist from the University of Buenos Aires School of Sciences and obtained a PhD in biochemistry from the same university. He then moved to England for a postdoc in Oxford, where he cloned the human fibronectin gene and found its alternative splicing. Since then, he has steadily made groundbreaking discoveries in the field of alternative splicing, going all the way from hardcore basic science to the application of his findings in the search for a cure of a neurological disease. And it is within this topic that he will be giving his seminar today, entitled Chromatin, Transcription, and Alternative Splicing in the Cure of a Hereditary Disease. Alberto is a plenary professor at the Department of Physiology, Molecular and Cellular Biology of the University of Buenos Aires School of Sciences and director of the Institute of Physiology, Molecular Biology and Neuroscience, and he has recently been named part of the board of directors of the Argentine National Research Council. The caliber of his work has been distinguished with several awards. To name a few, he was named International Research Scholar of the Howard Hughes Medical Institute, a Guggenheim Fellow, a Foreign Associate of the U.S. National Academy of Science, and a member of EMBO. He was awarded the prize Investigator of the Argentine Nation, granted by the President of Argentina, the Twice Prize in Medical Sciences, and the Diamond Connex Award as the most relevant scientist of the decade of his country in 2013.
Good morning, good afternoon, and good evening. Thanks, Belén, for your kind introduction. Thanks, Ron, for the invitation. We feel very proud of being the only hub in Latin America. And thanks, Janine, for the fantastic organization. Since I am also the coordinator of Sister Buenos Aires, I'll spend a minute on our institutions. The University of Buenos Aires is the biggest university in the country and one of the three biggest universities in Latin America, together with the Sao Paulo University in Brazil and the Autonomous University of Mexico. This is a free, no tuition fee institution. It has 13 faculties of schools, is owned by the state. It has nearly 300,000 undergraduate students. And in the history since 1821, we had 4 or perhaps 5 Nobel laureates, 2 of them in peace, 2 of them in science, Hussein was the one who found the connection between the hypophysis and the sugar metabolism, and Leloir was the discoverer of sugar nucleotides. And we can also count Cesar Milstein for the monoclonal antibodies, but he did most of his work in the UK. But we counted because he was an alumnus of University of Buenos Aires, and he was an accomplished scientist when he left the country in the 60s. So Hussein founded the Research Council in 1958. This is the Consejo Nacional de Investigaciones Científicas y Técnicas, that is the most important research and technology institutions, and it has 300 institutes nationwide, 10,000 investigators, 10,000 PhD students and postdoctoral fellows, and 4,000 technicians and administrative staff. So I will go now to my seminar. That is, the title is Chromatin Transcription and Alternative Splicing in the Cure of a Hereditary Disease. So my lab has been studying alternative splicing for decades, and we now know that it's more a rule than an exception. It's estimated to affect the expression of nearly 95% of human genes. It explains how a vast protein diversity can be achieved with a limited number of genes. And we know that mutations that affect alternative splicing sequences, called splicing enhancers or silencers, are a widespread source of human disease, both in cancer and hereditary disease. And the fourth issue is that alternative splicing regulation not only depends on the interaction of splicing factors with their target sequences in the pre-mRNA, but is coupled to RNA polymerase II transcription. And that has been the subject of my lab for the last 2 decades. So we know now that splicing is mostly co-transcriptional, and that splicing and alternative splicing are coupled to RNA polymerase II transcription. We found this many years ago by just swapping the promoter of a gene and finding that although the promoter is not transcribed, it affects alternative splicing decisions. So when we talk about coupling, it's different from co-transcriptionality. Co-transcriptionality is a need for coupling, but not necessarily everything that is co-transcription would be coupled. We talk about coupling when there is an influence of the splicing machinery or the transcription machinery on the splicing machinery, or the transcription kinetics on the splicing machinery. So the modes of coupling are not mutually exclusive, could be changes in Pol2 elongation, what we call kinetic coupling, or changes in the recruitment of processing factors to the RNA polymerase II itself, to its carboxy terminal domain, to chromatin, or to nascent RNA that we call recruitment coupling. And in this talk, we will focus on changes in Pol2 elongation. So we found many years ago that slow elongation promoted higher exon inclusion. And the model we were proving is the first come, first served, in which if you have an alternative exon like the one in green that has a weak 3' splice site that is competing with a stronger one downstream, if Pol2 goes fast, both sites are presented at the RNA level together to the splicing machinery, to the components of the spliceosome, and that would lead to skipping. But if Pol2 goes slow, then there is more time for the splicing factors to bind to the weaker site before the stronger site is made, and that would lead to inclusion. But in the case of the fibronectin exon we were studying as a model, the second intron is spliced first and the first intron is spliced second. That means that it's a window of opportunity for binding factors, but not strictly a problem of order of intron removal. So these exons in which slow elongation promotes higher inclusion are called the class 1 exons, and they account for 80% of the elongation-sensitive alternative splicing events. But there is a class 2 exons in which slow elongation can promote higher exon skipping, exactly the opposite. And this accounts for 20% of the elongation-sensitive alternative splicing events. And we were studying one of the cases in which we see that in this case, which is CFTR exon 9, if Pol2 goes normal, then there is no time for an inhibitor in yellow to bind to its target site in the pre-mRNA, and that would lead to exon inclusion. But if Pol2 goes slow, then the inhibitor has more time to displace the positive factors to bind to the target site, and that would lead to exon skipping. So I ask you to remember this case, because this is the case of the one we are studying in spinal muscular atrophy at the end of the talk. So the role of Pol2 elongation was confirmed by the use of a slow mutant of RNA polymerase II. Together with David Bentley, we made a human version of a slow mutant that had been described in Drosophila, and it accounted just for a single amino acid change in the large subunits of Pol2, an arginine for a histidine. So using Jeffrey Corden's strategy, we transfected cells with a mini gene, a reporter mini gene for alternative splicing, together with plasmids expressing either the wild type or the slow mutant of RNA polymerase II. But the fact is that these two constructs also contain a second mutation that made the enzymes, encoded enzymes, resistant to alpha-amanitin. So when we treat the cells with alpha-amanitin, the endogenous Pol2 is inhibited and later degraded, and both the mini gene and the endogenous genes are transcribed by the recombinant Pol2s, either wild type or slow. So in these examples, we see two classes of exons. In the case of fibronectin exon 33, the slow Pol2 promotes more inclusion. And on the right, in the case of TFTR exon 9, the slow Pol2 promotes skipping. And this was confirmed genome-wide by different labs, the Blenko lab, the Bentley lab, and the Cáceres lab, Blenko and Bentley in cells in culture, and Cáceres in mutant mice, knock-in mice with the slow Pol2. So what could change Pol2 elongation rate in a more physiological or pathological way that is not using a artificial mutant? Because this mutant doesn't exist in human cells, for instance. So it could be either modulation of Pol2 intrinsic activity via CTD phosphorylation, and we have studied this with Manuel Muñoz, and this is not what I'm talking about now, or changes in the template chromatin structure that could limit or facilitate elongation. So this is the effect of chromatin structure on alternative splicing that we found originally in the early 2000s together with Sebastián Cadener and Guadalupe Nogués in our lab. And after years of different investigations, we came to the conclusion that there is a model of alternative chromatin, alternative splicing, meaning that a same gene with different chromatin configurations, mostly intragenic chromatin configurations, can give rise to different elongation rates inside the gene intragenically, and that would affect this alternative splicing outcome. And in the case of the NCAM gene, we showed that, for instance, in certain conditions, when we produce neuron depolarization, there is a hyperacetylation of lysine 9 of histone H3, which is a permissive mark and opens the chromatin and allows for faster elongation. And in the case of the gene, that would go to exon skipping. But in the same gene, upon neuron differentiation, we can have another mark deposited or on the gene, which is the methylation of lysine 9 of histone H3. This is a silencing mark. It promotes chromatin compaction, and that chromatin compaction would promote slow elongation, and in this case, exon inclusion. So the opposite can occur with the class 2 exons. We could have an open chromatin promoting faster elongation that would be associated to exon inclusion, or we could have a compact chromatin with K9 methylation and slow elongation promoting exon skipping. So we investigated the effects of chromatin configurations in the disease spinal muscular atrophy. This is an autosomal recessive hereditary disease that causes defective motor neurons, poor innervation of skeletal muscle cells, and wasted skeletal muscle. And the cause of this disease is the lack or the short of a protein called SMN protein that is also involved in splicing. And this SMN protein is encoded by the SMN1 gene, but when this gene is mutated by deletions or point mutations, there is little protein. But this is not embryonic lethal in humans because there is a second gene, a paralog called SMN2. But SMN2 is in the same chromosome as SMN1, but it has 11 base pairs differences compared to SMN1. And these differences make an inhibitor, HNRMPA1, to bind better to a target site in the intron 7 and inhibit exon inclusion, of exon 7 inclusion. So the problem is the SMN2 gene is transcribed, is active, but it makes little protein because most of the splicing is directed towards skipping of exon 7. So we have about 20% of active protein and 80% of truncated protein that can be also degraded. So Adrian Krainer from Cold Spring Harbor, one of the sister institutes in this international seminars, developed an antisense oligonucleotide whose name is nusinersen, or the commercial name is spinRASA. And this is a heavily modified oligonucleotide that will try to change the splicing of the SMN2 gene. And what nusinersen does on the right here is to bind to the target site of HNRMPA1 and A2 and displace the negative factor, which promotes inclusion of exon 7. So the therapy is not based on the gene therapy of the mutated SMN1 gene, but on recovering the properties of the SMN2 gene to make 80% or more of the healthy protein. So Adrian is very famous, won every prize, and there are many movies in the web about the success of this therapy. I'm not going to show the movies, but just tell the case of one of the patients, Malcolm, who was born with SMA type 1, the most severe one, and that without treatment would have died in 6 to 11 months or survived prostrated for life with mechanical ventilation. But after 3 years, with continuous treatment with nusinersen and spinRASA, we can see a picture of Malcolm riding a tricycle. So the question was, is SMN2 exon 7 alternative splicing controlled by Pol2 elongation? And the answer is yes. We tried the slow polymerase, and we found that when transcription was driven by the slow polymerase, exon 7 inclusion was diminished, was reduced. So SMN2 exon 7 is a class 2 exon. But then more recently, we went to the genome-wide data published by the Bentley lab in a paper in 2014 and look at SMN2 exon 7 in the genome-wide sequencing. And we could corroborate not only that the slow polymerase could reduce exon 7 inclusion, but the fast mutant of RNA polymerase 2 could increase exon 7 inclusion. So clearly we know that SMN2 exon 7 is a class 2 exon, that when elongation is fast, it would have more inclusion. So we reasoned that chromatin opening should increase SMN2 exon 7 inclusion by promoting intragenic Pol2 elongation. So we decided to use histone deacetylase inhibitors like trichostatin A or TSA or valproic acid, VPA, that by promoting histone acetylation would open the chromatin and help exon 7 to be included. So spinRASA is the sequence on the top that we were using a modification of spinRASA, just having 2 more bases, which it was proved before that has the same efficiency as spinRASA. So we will call this ASO1, and we will use ASO1 every time in the next slides that we want to say that this is a nusinersen or spinRASA-like oligo. So you can see here in cells in culture, and the 2 bands on the gel on the top, the FL is the full length, and the delta 7 is the skipped exon 7, that ASO per se increases exon inclusion. TSA, that is a histone deacetylase inhibitor, also increases exon inclusion, but the 2 together are much more effective. And the same happens with valproic acid. ASO alone increases exon inclusion, valproic acid increases exon inclusion, but the 2 together are much more effective. And this is important because valproic acid is an FDA-approved drug that is used currently in the cure of epilepsy. So after having seen the results in cells in culture, we decided to go directly to do experiments in an SMA mouse model. So the fact is that mice have only one SMN gene. They don't have the backup gene. They don't have the SMN2. So if there are mutations in the 2 alleles of this gene, this is embryonic lethal. So some brilliant scientists from Taiwan decided to generate a model for SMA by generating a transgenic mouse in which they introduced the human SMN2 transgene into the genome of the mouse. So in this case, having the human SMN2 transgene, because it doesn't produce lots of the healthy protein, but it produces 20% of the healthy protein, the mice survive, it's not embryonic lethal, and they live for about 10 days after birth. So this is a good model to try reagents or drugs, because if they survive longer and they have weight gain and they are healthy, that means that we are waking up the good splicing of the human SMN2 transgene. So we did single subcutaneous injections of the nusinersen-like ASO1 and the HDAC inhibitor. And you can see here in pups 11 days old on the left that the ones injected with the ASO1 alone are smaller than the heterozygous, which has a bigger size. And on the right, that the ones injected both with the ASO1 and TSA are bigger, are the same size as the heterozygous. The same happens with 40 days old mice. You can see on the top, one injected with both the ASO1 and TSA, and on the bottom, the one injected only with ASO1 that is smaller. So when we measure the body weight gain, you can see that the mice treated with VPA die, so the curve is the green curve, but actually the ones treated with the ASO1 and VPA and the histone deacetylase inhibitor gain more weight than the ones treated with the ASO1 alone, and this is compared to the heterozygous that has a normal phenotype. But the most impressive data come now when we test the survival probability, and you can see that the ones injected with VPA alone die after less than 10 days, but the ones with ASO1 and the histone deacetylase inhibitor survive like 70% after 50 days compared to the ones injected only with the ASO1 with the nusinersen-like oligo that we have 15% survival after 50 days. So this is corroborated by the higher production of the SMN protein when we treat the pups with the 2 different reagents, with ASO1 and the TSA. So the combined treatment increases the amount of SMN protein in periphery organs like liver, kidney, muscle, spinal cord, brain, and heart. So we decided to test neuromuscular functions in the pups to see whether the combined treatment improved the performance of these animals. And you can see here that we were doing the surface writing. And this test, pups are placed on their backs and the time taken to write themselves is measured. You can see on the left that a pup injected only with ASO1 has difficulties to write itself, but the one injected with the ASO1 and the histone acetylase inhibitor immediately writes itself. So you can measure this, and you can see that the writing reference latency in seconds is very low by the wild type. Then the untreated mutant takes about 50 seconds. VPA alone doesn't improve this. ASO alone has a good performance, but the ASO and the histone deacetylase inhibitor have a better performance. And the second neuromuscular test is the grip strength. And in this case, pups are allowed to climb on a rough surface, and the angle at which they fall is measured. And you can see again that measuring the angle is much better with a combined treatment than with the treatment of the 2 drugs separately. So to finish, let me tell you that there are chromatin effects of the ASO that we were investigating that explain the effect of opening the chromatin that is, let's say, good for the effect of the ASO. So we had demonstrated many years ago in 2009 that if we transfect cells with intronic siRNAs in which the guide strand targets a pre-mRNA sequence of an intronic sequence that creates a chromatin compaction by creating hypermethylation of histone H3 lysine 9. This is mediated by the EGO1 protein in the nucleus and creates a roadblock for elongation that would affect alternative splicing decisions. So does the ASO1 create a roadblock to elongation? And the answer is yes. When we transfected cells with the ASO1, which is a single-stranded DNA, we could determine by chromatin immunoprecipitation there was a hypermethylation of the histone, not only at the target site, but along the entire SMN2 gene. And then the question is, does it create a roadblock to elongation? And the answer is also yes. When we transfect with the ASO1 oligo, we have very clear an accumulation of RNA polymerase II measured by chromatin immunoprecipitation, but this roadblock, this accumulation, is completely abolished when we treat the cells with also valproic acid. So it means that this roadblock would inhibit elongation and we have an opposite effect to the effect of the ASO1. So in fact, what happens is that the ASO1 is acting at two levels. At the RNA levels, as I told you, is displacing a negative factor and promoting exon inclusion, but at the chromatin level, it is promoting K9 methylation, promoting chromatin compaction, and acting in an opposite way, promoting exon skipping. So when we open the chromatin with VPA, we mitigate the effects of the chromatin compaction, we open the chromatin, and now the two mechanisms are acting together towards exon inclusion. So to conclude, we demonstrated that there is a kinetic coupling between transcription and alternative splicing, that slow elongation can promote either exon inclusion or skipping, depending on the particular alternative splicing event. We showed that change in the chromatin configuration we can change alternative splicing on the same gene in two different situations. And we are providing evidence for a combined therapy of spinal muscular atrophy with the ASO and chromatin opening drugs. So, and the mechanism for this cooperativity is that the nusinersen-like ASO1 has two opposite effects. The negative effect, which is the chromatin effect, is counteracted by opening the chromatin with histone deacetylase inhibitors. So most of the work, or almost all the work on the SMA issue was performed by the brilliant graduate student Luciano Marasco. But let me just say to finish that splicing control by transcription elongation seems to be important in many organisms. And our lab also studied this effect of the light and dark in plants. And just to finish two slides, we found that light controls alternative splicing in the nucleus of the plants using the chloroplast as a sensor. And what actually does is to send a signal from the chloroplast to the nucleus that affects elongation. And we showed that transcription elongation is fast in the day and slow at night, which means that a panoply of alternative splicing events are changing their proportions of messengers, the proportions of proteins during the day compared to the night because of changes in elongation. So I'd like to thank the people in my lab, not only the team related to chromatin and plants, but also to the other people working, for instance, in the effects of DNA damage and UV light on alternative splicing. And I'd like to thank my collaborators and the funding. I would stress that I have funding from HHMI from 2002 to 2017. And now I have great support from the families of the SMA patients, both FAME, the Argentinian families, and Cure SMA, the US families. So thank you very much, and I will be happy to take questions.
Thank you so much, Alberto. That was a fabulous talk. It's a very exciting story that you just shared with us. We do have a few audience questions, and while the audience is sort of thinking about the talk and getting their questions together, I do have one to start. Can you tell us a little bit about what is the normal physiologic function of the SMN protein, and specifically what is so critical about exon 7 that makes it absolutely necessary for healthy protein function?
Yes, thank you very much, Janine. The SMN protein is part of the SNRP cycle. It means it's part of the building of the SNRPs, which are the components of the spliceosome. So it's an essential protein for splicing to occur in all cells. We don't know why reduction in production affects specifically the motor neurons, but in the effects we are seeing with the combined treatment, we think are improving the general function of SMN in all organs. If exon 7 is not included, the protein doesn't work because it is truncated and it's finally degraded.
Thank you. I think we're going to turn to some of the audience questions now, and I'm going to call on some of the audience members, and if they would like to ask their questions, that would be great, and if not, I will go ahead and ask them. So we will start with Abhishek Bardhan. Abhishek, are you there?
Yes, can you hear me?
Yes.
Yes.
Yeah, I was just wondering, does this kind of chromatin structure controlled alternative splicing events have a role in in altered protein expression profile in cancer cells?
I guess so. So we don't have much evidence of specific roles of changes in chromatin in cancer cells, but I think it's a general, gene expression control mechanism that could affect alternative splicing in many pathological situations. I just would like to stress that not all alternative splicing events are sensitive to elongation, and not all of them are, of course, sensitive to chromatin, and that chromatin can also affect splicing, not only because of the control of elongation, but also it was shown that it would affect recruitment of splicing regulatory factors to the vicinity of the gene and the splice where the splicing event occurs.
Okay, thank you.
We also have a question from Eric Zhao. Eric, are you there?
Yeah, can you hear me?
Yes, go ahead.
Yeah, my question is, does the pausing of POL2 during elongation regulate the transcription coupled splicing?
Does the pausing of POL2 elongation, sorry?
Yeah, the pausing of POL2, polymerase
Yes.
during transcription elongation, does that regulate the transcription coupled splicing?
Oh yes, it does. I mean, pausing could be due to chromatin roadblocks or changes in chromatin structure, but also there is natural pausing by different reasons. There could be changes in POL2 phosphorylation inside the gene. And that pausing, if it is near or downstream of an alternative splicing event, could affect splicing, if that event is sensitive to elongation.
Mm-hmm. Yeah, thanks.
So we also have a question from Guihai Feng. Guihai, would you like to ask your question?
Hello, can you hear me?
Yeah, we can, but it's very faint. But you can go ahead and ask, and if we can't get it, I'll go ahead and read it for you.
Okay. Does VPA or HDAC inhibitor affected the normal splicing events of the other genes and lead to splicing of targets? Okay, thank you.
I couldn't guess it.
Let me get that. Let me read it from his written question. Guihai wants to know if the histone deacetylase inhibitors affect normal splicing events of other genes and possibly lead to splicing off targets.
Yes, of course. Histone deacetylase inhibitors can be pleiotropic, and of course, they will affect gene expression of many other genes. The only thing that we are in a way confident is that VPA has been used as an anticonvulsant in epilepsy and other neurological diseases. So it means that the secondary effects exist, but maybe not that important compared to the positive effects. And in this case, we think that because of what we saw in the mice, the positive effect will be probably overcome the secondary effects.
Okay, we have, I think we have time for another just one or two quick questions. This one is from Sam Banerjee. Sam, are you there? Would you like to ask? Okay, I'm going to go ahead and ask Sam's question. Sam wants to know if you have any idea what actually causes the change in the methylation pattern due to introducing ASO1.
Okay. In the case of the siRNAs, that was the original finding, we know that depends on Argonaute-1 roles in the nucleus that mediates the effect of the siRNA guide strand in recruiting the methylating enzyme. In the case of the ASO, we don't know. We are not sure yet whether it's dependent of Argonaute-1 or Argonaute-2, but these two Argonaute proteins have been shown to play roles in the nucleus, not only of mammalian cells, but also in yeast and other species. So we are investigating exactly the mechanism, but we think it's similar to the one we found with the siRNAs.
And we'll do one last question, and this one's from Alex Backer. Alex, are you there?
Yes, I am.
Go ahead.
Thank you.
Hi, Alex.
Hi, I'm a former student of Alberto, and I have to say that having studied biology at MIT, Harvard, and Caltech, Alberto's course of molecular biology was the best course in biology I have ever taken. And he's formed, he hasn't said this because he's modest, but he's formed generations of Argentinian students. So that's a big merit. My question is, what are the implications of alternative splicing versus different genes as mechanisms for evolution? And what's the multiplicative factor of how many different forms of genes are there because of alternative splicing compared to how many there would be without?
Well, as I mentioned, thank you, Alex, you made me blush. But as I mentioned, 95% of human genes are subjected to alternative splicing. Part of it is noise, but that noise has an evolutionary function that is to allow for selection of cases in which having one or other alternative splicing option has an adaptive value. So we think that having 23,000 genes, about that, human cells, mammalian cells can make more than 100,000 or 200,000 different protein variants. And that is a huge advantage in evolution, because you have to bear in mind that invertebrates have a similar number of genes, like C. elegans, 19,000, but the number of protein variants is much smaller.
Great. Thanks, Alberto.